Today, I would like to extend our gratitude to and introduce Dr. Fanny Mann. Dr. Mann is a neurobiologist who, since 2012, has led her own research team within the Centre National de la Recherche Scientifique and currently serves as research director of the Marseille Developmental Biology Institute in France. Today, we have the opportunity to learn something new, at least to me, and I am sure also for many of you. Indeed, we are bridging neurobiology and tumor biology in a seminar entitled Visualizing and Understanding Neuronal Plasticity in Pancreatic Cancer Development. Let me say that I'm really curious about the content of this presentation. So please, Fanny, the screen is yours. Thank you so much again.
Thank you very much for this introduction and for the invitation to present our work. So can you see my screen?
Yes, we can see it.
Yes, clearly.
Good, thank you. So I'm not going to talk about liver, but about pancreatic cancer. But I believe that some of the mechanisms we have identified might be also at play in liver cancer. So I'm a developmental neurobiologist, and my group originally specialized in studying how these beautiful patterns of neuronal projections develop during embryonic life. And this is a very complex process during which the newborn neurons extend an axon that navigate often over long distances to reach their proper targets. And during this navigation of the axons, they meet and they integrate information provided by several molecular cues expressed along the path. So these molecular cues belong to the large families of semaphorins, lead, netrins, or ephrins, and they can steer the growth of axons through either attraction or repulsion. And my group has contributed to, with others, in identifying some of these axon guidance cues and how they control the precision of the establishment of these neuronal projections. So once these neuronal projections are established, they have to be maintained through adult life, and this is important for the proper functioning of the nervous system. But these adult neuronal projections, they retain some degree of structural plasticity that allow them to adapt and to respond to some stimuli that may vary depending on experience in life, but that can also arise from disease, and in particular from the development of malignant tumors. And it is now more and more established that solid tumors are indeed innervated. And there is evidence for the presence of nerves or nerve fibers in several types of cancer, including head and neck cancer, pancreatic cancer, prostate, and also breast cancer. So this raised interesting questions about the origin of these intratumoral nerves and whether the tumor might be able to co-opt some of the developmental mechanisms of axon growth and guidance in order to attract new nerves and to promote their own innovation. And this hypothesis is supported by the findings that axon guidance molecules that contribute to the establishment of neuronal projection during embryonic development are often re-expressed in cancer. And there is a lot of research that have been done over the last 2 decades to identify function for axon guidance molecules in tumors. And we know that molecules such as semaphorins, natrins, or ephrins play important function in the regulation of the tumor ecosystem. They can act directly on the regulation of the cancer cells themselves, modulating their proliferation, survival, or migratory properties. Axon guidance molecules can also contribute to the recruitment of immune cells and in the regulation of the activity of these immune cells. And they have also an important function in the regulation of tumor neoangiogenesis. But the paradox is that after about 2 decades of research on the role of axon guidance molecules in cancer, we still don't know whether they contribute to tumor innovation. So we have chosen to address this question in the context of pancreatic ductal adenocarcinoma. The reason why we choose this type of cancer, it's because we know that the pancreas is an organ that received an abundant nerve supply that supports its function of nutritional assimilation and glucose homeostasis. It's also a type, one of the first cancer in which the presence of nerves have been reported. And this, the first observations were made about 15 years ago by a group of clinical researchers in Germany. And they reported both an increase in nerve density and a hypertrophy of the nerves in sections of pancreatic cancer compared to normal pancreas from healthy human donors. And in this initial study, the degree of neuronal changes was associated with a high severity of the pain reported by the patients. However, there was no association with overall patient survival. And since then, 2 separate studies have reported a relationship between the presence of nerves in tumors and patient survival, but their findings are in fact contradictory. So it is clear now that pancreatic tumors are innervated. This has been shown both in human samples and in animal models. However, on a clinical point of view, the significance of the presence of this nerve and their prognostic value is still debated. But it seems that the presence of nerve is usually associated with advanced malignancy. So the question that we are interested in is to understand what is the origin of these intratumoral nerves. And the observation of the presence of high density of nerve, of a higher density of nerves in tumors compared to healthy tissue or adjacent tissue suggests that cancer development may lead to an expansion of nerves. And the current model is that pancreatic tumors may be able to actively recruit nerves from surrounding tissues through the secretion of axonogenic factors, which are still not identified. And these axonogenic factors will cause the nerves to branch out and to grow within the tumor microenvironment. And in support of such a model, several studies, in fact, have shown that if you co-culture neurons from the peripheral nervous system with pancreatic cancer cells, you will observe a higher growth, an increased outgrowth of the neurons in the presence of pancreatic cancer cells compared, for example, to normal pancreatic ductal cells. So such a mechanism of recruitment of new nerves will enable the growing nerves to establish interactions with different cells in the tumor, cancer cells, or also blood vessels or immune cells. And this dialogue may have a function in promoting tumor development. So if so, understanding what are the mechanisms that attract the growth of neurons and induce their plasticity could lead to new approaches to cancer treatment. So in my talk today, I will present you what we have learned in my group about the mechanism of neuronal plasticity in pancreatic cancer. And my talk will be divided into 3 parts. So first, I will show you the results we have on the mapping of pancreatic cancer innovation. In the second part, I will show you some results about the role of axon guidance molecules in this cancer-induced neuroplasticity. And in the third part, I will show you what we have learned about the role of nerves in the control of pancreatic tumorigenesis. So our primary goal was to develop new methods in order to map the innovation of pancreatic cancer. So why new methods? It's because to date, what I've presented you is the analysis of tumor innovation has mainly relied on measuring the number and the size of nerves on histological sections. But tissue sectioning has limitation for this type of analysis. First, because a section will represent the innervation of only a very small region of the organ or of the tumor. And second, because it's impossible on sections to trace the origin of these nerves. And very importantly, on sections, you only see the large nerve bundles. And these nerves will eventually target, will innervate the target tissue, but the very fine nerve endings in the target tissue are very difficult to identify on histological sections. So we have, for the moment, no knowledge of the distribution of the nerve endings in the tumor microenvironment. So to get around these limitations, we have decided to map the innervation of pancreatic tumors using a combination of tissue clearing techniques and light sheet fluorescent imaging. So we have adapted this technique to wall mount preparation of mouse pancreas. And you see here a mouse pancreas. We usually keep the spleen attached to the specimen, and this is to allow us to orient the head and the tail of the pancreas. And this is important because these 2 regions of the pancreas are not innervated the same way. And we have so clear pancreas from either wild-type mouse or transgenic mouse models that spontaneously develop pancreatic adenocarcinoma. So in the lab, we have studied 2 oncogenic KRAS-based transgenic mouse models, the KIC and the KPC mouse models. And we have observed the same neuroplastic changes in the 2 models. So the results I will present you today are from experiments performed in the KIC model. So an important first step for our study was to be able to identify the cancerous lesions in the pancreas that were made entirely transparent. So this can be achieved by staining the tissue with an antibody against epithelial markers, such as cytokeratin 19. So this allows to see here the tumors. However, what we found is that pancreatic tumors can also be distinguished without specific staining, just by simply using the autofluorescent signal of the tissue. And the tumors usually has a lower signal intensity compared to the adjacent tissue. So we have taken advantage of this property to develop deep learning-based methods that allows us to automatically detect and reconstruct in 3D all the tumor nodules that develop in a pancreatic specimen. So with this method established, then we looked at the nerves. So this is a scheme of the innervation of the pancreas. To make it simple, the pancreas receives extrinsic innervation from sensory fibers, both vagal and spinal sensory fibers, which are depicted in green on this scheme. And it also received efferent motor projections from the autonomic nervous system. And the autonomic nervous system is composed of three main branches: the sympathetic, parasympathetic, and enteric nervous system. So today in my talk, I will talk exclusively about the sympathetic nervous system. What you have to know is that the sympathetic neurons that project to the pancreas have their cell bodies located outside the pancreas in a group of ganglia called the celiac superior mesenteric ganglia. And these are adrenergic neurons. So they produce, their main neurotransmitter is norepinephrine, which acts on adrenergic receptors expressed on their target cells. So we can visualize the sympathetic inputs to the pancreas by staining the neurons with an antibody against tyrosine hydroxylase, TH, which is the main limiting enzymes in the biosynthetic pathway of norepinephrine. So here I'm showing you the result of an anti-TH staining on a mouse pancreas. So on the left, you see the entire reconstruction of the sympathetic nerves in the wild-type mouse, and on the right, it's in the KIC mouse. So in the KIC mouse, the tumor nodules have been segmented and reconstructed in blue. And the sympathetic nerves located inside the tumor nodules have been colored in yellow. And the sympathetic nerves outside the tumors are shown in red. So consistent with what has been shown already in human samples, we have found a higher proportion of sympathetic nerves within the tumor nodules in the KIC mouse models. However, there were clear regional differences in the innervation of the tumors. For example, the tumors that develop here in the head of the pancreas, which is a region that already contains a lot of nerves in the control mice, they have a correspondingly high presence of intratumoral nerves. In contrast, the tumors that arise in the tail of the pancreas have few intratumoral nerves, and as you can see, the tail of the control pancreas also contains few sympathetic nerves. In addition, when we trace in 3D, when we trace the course of these intrapancreatic intratumoral nerves, we found that some of them clearly correspond to segments of nerves that are already present in the healthy pancreas. So these observations suggest that the nerves in the tumors are not newly formed, but they might be pre-existing pancreatic nerves that have been passively engulfed into the tumor. And in support of this idea, we have quantified the total amount of sympathetic nerves in each pancreatic sample, and we haven't found any significant difference between the control and the KIC pancreas. So this confirmed that there is no new nerves formed during the development of pancreatic cancer, but just that the tumor engulfs existing nerves. So next, the next step was to study the terminal endings of the sympathetic nerves in the pancreas. So the large nerves we have seen so far that enter the pancreas, at one point, they will defasciculate into individual axon fibers to innervate their targets. So sympathetic neurons in the pancreas, they innervate the pancreatic islets. And we know that sympathetic signaling, it's important to promote, for example, the secretion of glucagon and to inhibit the secretion of insulin. But the sympathetic nervous system also densely innervates the exocrine part of the pancreas. And sympathetic signaling in the exocrine pancreas is also important to inhibit the secretion of the digestive enzymes through the regulation of the blood flow. And so here we can zoom on the pancreatic lobule to have a look at this terminal innervation of the exocrine pancreas. And you see that the parenchyma of the pancreas is full of this dense and ramified network of individual sympathetic axons. Interestingly, when we look at this terminal innervation of the exocrine pancreas in KIC mice, we observe regions like this one, hotspots of sympathetic innervation that we don't see in control organs. And these hotspots of sympathetic innervation correspond to very dense regions of sympathetic innervation that have grown and branched around small epithelial lesions, which in fact correspond to early precancerous lesions that we called pancreatic intraepithelial neoplasia, or PANI. So we were very interested with this finding that suggests that that neural remodeling related to the development of pancreatic cancer does not involve the major sympathetic nerves, but instead involve a local remodeling of their terminal endings within the exocrine tissue. So to extend and to confirm these findings, we have performed a reconstruction of the sympathetic terminal innervation across the different stage of cancer development. So from acinar to ductal metaplasia to precancerous pancreatic lesions and invasive PDAC. And what we have observed is an increased axonal density in all lesions compared to the asymptomatic pancreatic tissue. And this hyperinnervation was particularly prominent in the early metaplastic and precancerous lesions. Next, we asked how the sympathetic axons in these lesions might interact with other cell types in the microenvironment, and in particular with blood vessels, because we know that sympathetic neurons innervate blood vessels and play an important function in the maintenance of vascular physiology. So we stain and reconstruct the axons in red and the blood vessels in green. And the surface of contacts between the two networks were extracted, and they are shown in yellow in the bottom panels. And what we found is that in the healthy tissue, the sympathetic axons and the blood vessels are perfectly well aligned. But in all precancerous and cancerous pancreatic lesions, the contact surface between the axons and the blood vessel were much smaller and more fragmented. So we have quantified all these changes in axonal morphology and interactions with the blood vessels, and our results have shown that both precancerous and cancerous pancreatic lesions display aberrant patterns of sympathetic innervation. These aberrant patterns of innervation are characterized by an increased axonal density and an increased axonal branching. And we discovered that also that unlike what happened during development, where sympathetic axons follow blood vessels, during cancer-induced neuroplasticity, the sympathetic axons remodel independently of the blood vessels. So this suggests that in these lesions, the sympathetic neurons might have another function than the one they have in the physiological condition. And I will come back to this idea later in my talk. So at the conclusion of this first part of my talk, we have found that two distinct and coexisting mechanisms can explain the presence of nerve fibers in the microenvironment of tumors. First, a passive engulfment of the existing nerve bundles. And second, an active sprouting of the sympathetic axon terminals and this sprouting is already occurring before at pre-malignant stage, so before the onset of cancer. So our next question was to understand what is the molecular mechanism underlying this sympathetic axon sprouting. So to answer these questions, we have used a mouse model of chronic pancreatitis induced by repeated injections of cerulean. So this model produces metaplastic pancreatic lesions that resemble the earliest lesions in the development of pancreatic cancer. So this allows us to observe the very initial events in the induction of the sprouting of sympathetic axons. And the advantage of this model also, it's since it's not a genetic model, it can be integrated with genetic inactivation models, and this allows us to mechanistic questions to be addressed more easily. So the metaplastic lesions that are induced by chronic pancreatitis are characterized by an ectopic expression of ductal markers in metaplastic pancreatic cells, by an infiltration of macrophages in these lesions, and by an increased innervation by sympathetic axon terminals. So we observe the same induction of sympathetic axon sprouting as we have seen in the KIC mouse model. So we use this model to investigate the expression of receptors for axon guidance molecules on the sprouting sympathetic axons. And one receptor for axon guidance molecules show a very interesting profile of expression. And this receptor is called DCC. It stands for deleted in colorectal cancer. And this is a transmembrane receptor which is expressed on the surface of sympathetic axons during embryonic development, and which is really important. Its function is important for sympathetic axons to innervate arteries. After the development, DCC expression goes down, and it is barely detectable on the sympathetic axons in the adult, and we found no expression on axons in the healthy pancreas. However, after induction of chronic pancreatitis, DCC receptors is upregulated on the sprouting sympathetic axons. We have confirmed this upregulation of DCC by RNA-scope in situ hybridization performed on the celiac superior mesenteric ganglia, so the ganglia that contain the cell bodies of the sympathetic neurons projecting to the pancreas. So to know whether this upregulation of DCC was important for sympathetic axon sprouting, we've used an inducible conditional approach in the mouse to delete DCC from adult sympathetic neurons, and just prior inducing chronic pancreatitis. And we have found that the loss of DCC on sympathetic axons completely inhibits the hyperinnervation of the metaplastic lesions. So demonstrating that DCC function is important for sympathetic axon sprouting. So DCC, as I told you, is a receptor for an axon guidance molecule, which is called netrin-1. So we look at the expression of the netrin-1 ligand in the model. And in the adult pancreas, the protein, netrin-1 protein, is not expressed. But after cerulean injection, netrin-1 is expressed by two types of cells, the metaplastic pancreatic cells, and also by populations of inflammatory macrophages that infiltrate these metaplastic lesions. So this suggests that netrin-1 might be the signal that triggers sympathetic axon sprouting. So we first tested this idea in vitro. So we dissect out the celiac superior mesenteric ganglia and we dissociated the neurons and we put them in culture. In culture, they re-express DCC, and we found that when we apply recombinant netrin-1 to the culture medium, this induced an increased growth of the neurites, of the sympathetic neurites. And we can block this growth-promoting activity of netrin-1 using a netrin-1 blocking antibody, whereas the isotype control antibody had no effect. So this suggests that netrin-1 can stimulate the growth of DCC expressing sympathetic neurons. So we tested this idea directly in vivo by treating the mice with netrin-1 blocking antibody at the same time that we induce chronic pancreatitis. So here are reconstruction of the sympathetic innervation of the metaplastic lesions in the control and the anti-netrin-1 treated lesions. And we found that blocking netrin-1 decreased both axonal density and axonal branching in the metaplastic lesions. So we wanted to confirm these results and also using a genetic approach and also to identify the relevant source of netrin-1 for the sprouting of sympathetic axons. So for this, we selectively silence netrin-1 either in macrophages using a LISM-3 mediated recombination or in pancreatic cells using a PDX1-3 recombination. And that after cerulean treatment, the loss of netrin-1 only in pancreatic cells, but not in macrophages, significantly inhibits sympathetic axon sprouting. So what we think is that the metaplastic reprogramming of acinar cells into epithelial-like cells induce the expression of netrin-1, which act on the DCC expressing sympathetic nerve terminals to induce their local growth and sprouting. So next, we wanted to know whether this this netrin-1 DCC pathway was also important to induce cancer-associated neuroplasticity that we have observed in the KIC mouse model. So we treated the KIC mice with the netrin blocking antibody or the isotype control antibody, and we treated them from 4 to 6 weeks of age. So at the time of the onset of precancerous Pannin lesions and their progression to PDAC. And here are example of TH staining in precancerous Pannin lesions, and on the right side you have the reconstruction. So in the mice treated with a control antibody, we found what we have already observed is a hyperinnervation of the Pannin lesions. But when we treat the mice with a netrin-1 blocking antibody, then we found a strong reduction in the innervation of these Pannin lesions, while the innervation pattern of the adjacent tissue was not affected. So this shows that Netrin-1 is important for the innervation of the precancerous lesions. And this decrease in innervation we observe when we block Netrin was also associated with an increase in the formation of Pannin lesions in the treated pancreas. And this increase in the number of Pannin lesions was also associated with an increase in the ratio of high-grade lesions in the pancreas. So together, our results indicate that Netrin is important to stimulate the sympathetic innervation of Pannin lesions, and that maybe through this innervation, Netrin also inhibits the formation of Pannin and their progression toward cancer. So, so as a conclusion for this part of my talk, so what we have shown is that the sprouting of nerve terminals is induced by the re-expression in the pancreas of a developmental axon guidance molecule, Netrin-1, that induce sympathetic axon sprouting, and this sprouting might be important to limit the formation of Pannin and their progression to PDAC. So the Netrin might be part of a defensive mechanism at this stage of the development of the disease that would inhibit the cancer progression. So we wanted to directly test indeed this potential protective activity of the sympathetic nervous system in pancreatic cancer. So to do this, we have performed an ablation of the sympathetic nervous system using a chemical approach by injecting the neurotoxin called 6-hydroxydopamine, 6-HGA, in KIC mice. So here again, we treated the mice early, so we performed this sympathectomy before the onset of precancerous lesions. And what we observed is that the removal of the sympathetic innervation accelerated tumor growth. It also induced the spreading of the tumors to the liver and to the peritoneal cavity. And that was very striking because the KIC model usually does not lead to metastatic disease. And we also observe a decrease in the overall survival of the KIC mice after sympathectomy. So again, these data support the idea that the sympathetic nervous system might have an antitumorigenic effect at early stage of pancreatic cancer development and progression. These results were very surprising because in other type of cancer, sympathetic nervous system has been found to promote the initiation of cancer, in particular in prostate cancer. So we wanted to confirm these results in another model. So we use a luciferase expressing mouse pancreatic cancer cells derived from the KPC mouse model, and we graft these cells into the pancreas of syngeneic mice. We follow the growth of the tumor by bioluminescence imaging, and we fed the data to a mathematical Gompers model, from which we estimated two main parameters. The first parameter is log B, which corresponds to the logarithm of the highest bioluminescence level. In other words, it's the plateau reached by the curve. And the second parameter of this Gompers model is A, which represents the rate at which the growth curve reaches its maximum. So the estimated log B and A parameters are shown on this 2D graph. for the control mice and the sympathectomized mice. What you can see that the two groups are clearly separated from each other, with the tumors in the sympathectomized mice growing longer to reach a higher size. Again, confirming that the sympathetic nervous system exerts a protective function against tumor growth. However, when we perform the same experiment, or we graft the same cells, but that time in atymic nude mice, we found that the two groups were no longer separated. So this tells us that this accelerating effect of the sympathectomy on tumor growth requires a host intact immune system. So to begin to understand what was the mechanism underlying this effect, we use a digital spatial profiling technology to profile the immune infiltrate in KIC tumors and compare the immune infiltrate in the control and denervated tumors. And we found that a few immune markers were upregulated in the denervated tumors, and we decided to focus on this upregulation of the CD163 marker. So CD163 is a receptor that is expressed exclusively on tumor-associated macrophages, and its high expression has been correlated with a poor outcome in pancreatic cancer. So by immunohistochemistry, we have confirmed that in denervated KIC mice, we have an increase in the proportion of CD163 macrophages in the tumors. We have no difference in the total number of macrophages, but the population of CD163 positive macrophages is increased. And then we ask whether the adrenergic activity of the sympathetic nervous system might be directly able to regulate the macrophage identity. So for this, we FACS sorted the CD163 positive macrophages from the pancreas of the mice, and we exposed these macrophages to different concentration of norepinephrine, which is the main neurotransmitter of the sympathetic nervous system. And we found indeed that norepinephrine was able to decrease the expression of CD163 in pancreatic macrophages when it was used at high concentration. So this suggests that maybe the sprouting of sympathetic axons might allow to induce high local release of norepinephrine that might contribute to limit the number of these macrophages in pancreatic lesions. And finally, we wanted to know whether this increase in CD163 positive macrophages was directly responsible for the poor outcome of the sympathectomized KIC mice. So to answer this question, we used nanolipids loaded with doxorubicin and vectorized with anti-CD163 antibodies. So these nanolipids, when they are injected in the KIC mice, they exert cytotoxic activity specifically kill the CD163 positive macrophages. And we achieve a 70% reduction in the amount of CD163 positive macrophages in the KIC tumors. And we found that this ablation of this population of macrophage was sufficient to rescue a normal survival of the sympathectomized KIC mice. So in conclusion of this presentation, I've shown you that the observation of nerve or nerve fibers in the microenvironment of a tumor does not necessarily mean that these are new nerves. It can be pre-existing nerve bundles that have been engulfed into the tumor. What is important more than looking at the nerve, it's important to look at their terminals which is a region where they secrete neurotransmitters and neuropeptides. And we have seen that these nerve terminals actively sprout both in precancerous lesions and in pancreatic adenocarcinoma. And we have found that this remodeling, this plasticity of the sympathetic nervous system is induced by the reactivation of a developmental axon guidance pathway. And this nervous system, instead of being associated to blood vessels and to participate to vascular tone, is not associated with blood vessels in the lesions and might have another function, which might be an immune regulatory function that is important to mediate its antitumorigenic effect during the development of pancreatic cancer. And to finish, I'd like to acknowledge all the people from the lab who have done the work I presented, and our collaborators that have really been crucial for us to develop this project at the interface between cancer, immunology, and neuroscience. And thank you very much for your attention.
Thank you, Fanny, so much for this really nice presentation.